Hi everyone, my name is Alessandra Norris. I'm from UF in the lab of Dr. Kopinski, and today I'll be talking about uncovering a novel mechanism for intramuscular fat. So I first wanted to introduce three possible fat, there we go, no one move. I first want to introduce three possible fat depots in the human body. Subdermal, visceral, and intramuscular. Subdermal is located throughout most of the human body, mainly underneath the skin. So if we see in the abdominal area, the subdermal fat would be located on the exterior part of the abdominal wall. However, visceral fat, which is located in the abdominal area, is located inside the abdominal wall. Now, if we look at one of the extremities, like a leg, we can see that we have subdermal fat around the muscle, whereas intramuscular fat, or intramuscular adipose tissue, IMAT, is located within muscle fibers. So if we look closer, we can see that fat cells, shown here in red, are located between and surrounding muscle fibers here in cyan. Now, in healthy muscle, there is very limited amount of IMAT, and during regeneration, there's also not that much IMAT formation. However, this process can go awry, particularly in disease, leading to an increase in IMAT and decrease in muscle mass. And this is characteristic of neuromuscular diseases and muscular dystrophies, spinal cord injuries, and age and obesity. And this increase in IMAT ultimately leads to muscle dysfunction, metabolic, and mobility dysfunction. So I'm primarily interested in what signaling cues goes awry that leads to IMAT formation in order to regulate or control this. So the Hedgehog Pathway has been extensively studied during embryogenesis, where it regulates and dictates stem cell fate, leading to limb, bud, and digit formation, neural tube formation, craniofacial development, and left-right axis of the human body. So does hedgehog play a role during adult skeletal muscle too? So we and others have shown that pharmacological and genetic activation of the pathway leads to a repression in fat and enhancement of muscle regeneration. However, there are many unanswered questions, such as what cell types are responding, what are the downstream targets, and can it be used therapeutically? But I first wanted to start answering a more fundamental question, which is which ligand is involved. And this is because physiologically there are three proteins that activate the pathway: sonic, desert, and Indian, and there has been debate whether sonic or Indian are the endogenous proteins that are regulating this pathway. So we turn to single-cell RNA-seq in collaboration with Dr. Crosgrove and his student David McKellar in Cornell University, and we queried around hundreds of thousands of cells after a cardiotoxin or notoxin injury at different time points. And we looked at the different expressions of the ligands. And while we saw that sonic and Indian barely had expression, we saw a robust expression in desert hedgehog, particularly in the endothelial and the neural cells. So what does desert hedgehog do? So to answer this, we first looked at intramuscular fat formation. After a cardiotoxin injury, 21 days, we harvested the tissue. And in a control setting, we see that there is, where desert is present, the hedgehog is on. And in a control setting, we see limited amounts of fat depicted here in green, whereas we used a genetic mouse model where we removed desert, rendering the pathway off. And 21 days after cardiotoxin injury, we see a significant increase in intramuscular fat. So while turning the pathway on represses fat, if we remove the ligand that endogenously turns the pathway on, this renders it off, allowing fat to form, suggesting that desert acts as an adipogenic brake. So what about muscle? We looked at cross-sectional area of single muscle fibers, and here I've color-coded muscle fibers according to their size. So darker red is smaller fibers, whereas yellows and whites are bigger fibers. And 21 days after cardiotoxin injury, we see a significant reduction in cross-sectional area, suggesting that desert is necessary for myofiber regeneration. So in summary, I have shown that desert is the endogenous ligand that is activating the pathway, leading to a repression in fat and an enhancement of muscle regeneration. And before I leave, I want to answer just one more question, which is what are the possible downstream targets? Previous research from our lab suggests that it could be TIMP3, which is a tissue inhibitor of metalloprotease 3. And in the desert knockout animal, if we look at 3 and 7 days after cardiotoxin injury, we see that TIMP3 is down. So if we remove desert, TIMP3 is down, suggesting that there could be a correlation. So to test whether it is TIMP3, I used a small molecule called badimastat, which is a TIMP3 mimetic. So after cardiotoxin injury, we gave badimastat 0, 2, and 4 days and harvested 7 days after cardiotoxin injury. And in a vehicle setting, we see that fat is increased and muscle is decreased, and this is no new news. However, upon badimastat administration, we see a rescue in the phenotype of fat comparable to control levels, whereas with muscle, we do not see a rescue. So this suggests that TIMP3 is specifically inhibiting IMAT infiltration, and there could be another downstream target that also regulates muscle regeneration. And so with that, I want to thank you for your attention, my lab, Compiki Lab, committee members, collaborators, and my T32 training grant.